Welcome to this explainer. Today, we're diving into something absolutely fascinating. We are going to explore a recent study that outlines the precise, step-by-step molecular construction of a completely novel antibacterial agent. That's right, we're taking a trip directly into the chemistry lab to see how scientists are literally building the future of medicine from the ground up. To kick things off, we really need to ask ourselves a fundamental question. How do chemists actually invent new medicines from scratch to fight against rapidly evolving diseases? Well, it isn't magic, it's molecular architecture. And today, we're going to explore one specific, highly successful blueprint. Here's a quick look at our roadmap for today. 1. The need for new cures. 2. Meet the shift basis. 3. Synthesizing the molecule. 4. Inside the crystal structure. And 5. Antibacterial test results.
All right, let's jump right into Section 1, the need for new cures.
The stakes here literally couldn't be higher. Millions of lives are lost globally each year to bacterial infections. And because bacteria are constantly evolving, constantly developing resistance to our current drugs, there's a massive, urgent scientific push to discover entirely novel, effective therapeutic agents. We're basically in a constant arms race, and scientists are desperately searching for new chemical weapons to add to our medical arsenal. Which brings us to Section 2, Meet the Schiff basis. Developed way back by German chemist Hugo Schiff in 1864, these compounds are formed by condensing primary amines and aldehydes. But here's why they actually matter to us today. These compounds act like incredibly versatile structural scaffolds. Think of them kind of like the highly adaptable chassis of a car. By tweaking the chemical attachments, scientists can optimize them to produce completely new drugs with potential antibacterial, anti-inflammatory, or even anti-cancer properties.
Now for the really fun part. Section 3, synthesizing the molecule.
Think of this process just like molecular Lego. We start by snapping together two very specific building blocks. We take a compound called 1,2-diaminotoluene and drop it into a heated refluxing solution of Furil in an ethanol bath. We let that simmer for about an hour until a distinct brown color appears. And finally, the researchers filter and isolate the product, which actually takes the form of pale brown needle-like crystals.
And this perfectly illustrates the exact chemical recipe we just described. We have Furil combining with 1,2-diaminotoluene, shedding water, and creating our complex new chemical weapon. This simple atom-efficient process resulted in a pretty impressive 65% yield of our new compound, which researchers observed melting at exactly 160 degrees Celsius.
Moving right along to Section 4, Inside the crystal structure. So, using X-ray crystallography, scientists mapped every single atom to see the brand new architecture we just built. The real star of the show here is the crucial new imine linkage. That's the specific double bond between the carbon atom of the Furil and the nitrogen atom of the Diamine. That linkage right there is the signature of our Schiff base. The crucial point derived from this mapping is the molecular blueprint itself. The data reveals that this new molecule forms a monoclinic crystal system. And what's absolutely fascinating is that it's highly asymmetric. It possesses no mirror planes, no stereo centers, and no inversion centers whatsoever. It is a completely unique geometric shape. And here is how those unique shapes stack together in the real world. We can see how 8 of these individual molecules pack together into a linear chain along a single axis, stabilized gently by van der Waals forces. We can also build a volumetric 3D model of our molecular weapon, which shows the actual physical space its electron clouds occupy. But of course, chemists don't just look at a crystal and guess that they built the right thing. They test it rigorously. Taking a look at the powder X-ray diffraction pattern, the sharpness and position of the specific peaks prove the compound's extraordinarily high crystalline purity. It reveals an incredibly fine average grain size of just 3.83 nanometers.
Furthermore, they use infrared spectroscopy to verify the specific bonds. Now, instead of getting bogged down in every single number on the spectrum, just look at the sharp peak labeled 1697 inverse centimeters. That specific dip is essentially the smoking gun. It undeniably proves that our critical carbon-nitrogen double bond, the very heart of the Schiff base, successfully formed. The chemistry worked absolutely perfectly.
Finally, we reach Section 5, Antibacterial test results. It's time for the real-world battle. The researchers set up the battleground using an in vitro agar well diffusion test. They took 300 micrograms per milliliter of our newly synthesized compound and pitted it against 4 notoriously stubborn bacterial species, including Staph and Salmonella. To know if it was actually effective, they compared its killing power directly to a well-known, highly effective standard reference drug, doxycycline. Looking directly at the results for Staphylococcus aureus, in the agar dish, our new molecule created an 18-millimeter zone of inhibition. That's basically a clear circle where the bacteria were entirely eradicated. Now, doxycycline created a 25-millimeter zone. So while 18 millimeters isn't 25, it is a highly respectable, moderate level of antibacterial activity for a compound straight out of its very first synthesis.
In fact, when we look at the full data across all 4 bacterial strains, we see real consistency. Our compound scored 18-millimeter clearances against both Staph aureus and Salmonella typhi. It also successfully inhibited E. aerogenes and B. subtilis, proving its broad-spectrum viability across some very different strains of bacteria.
So, what is the main takeaway here? While overall our new compound showed low to modest activity compared to a fully developed commercial pharmaceutical like doxycycline, this fundamentally proves our concept. Our molecular Lego approach successfully built a functional new antibacterial scaffold that actually kills dangerous bacteria.
As the authors confidently state in the paper, this compound could absolutely be a better drug candidate for antimicrobial infections. It serves as a vital, proven stepping stone in the ongoing scientific pursuit of novel antibiotics. By slightly modifying this newly proven scaffold, the next iteration could be even more powerful than doxycycline.
Which leaves us with one final, exciting thought to wrap up this explainer. If we can synthesize a brand new, functional antibacterial agent from scratch using just 2 basic ingredients today, what incredible, life-saving combinations remain undiscovered for tomorrow? The possibilities in the chemistry lab are quite literally endless. Thanks for joining me to explore this fascinating molecular journey, and keep seeking out the science that shapes our world.